in the 1950s, ECOG was developed and it was then FDA approved in the mid-70s. This is the only device of its kind that's actually FDA approved. The biggest problem with ECOG at the moment is that it still has to remain wired. That means that there's, at the exit points, there are a high risk of infection or continued bleeding. The universities of Utah and Iowa later made their own chronic electrode arrays, but these were extremely rigid. And so a small bump to the wearer's head could cause it to shift and cause irreversible damage to the brain. We aim to utilize this previous research and to build upon it. We want to create an electrode array that's very flexible, but still completely implantable. Our first question then is, where exactly do we want to place this electrode?
The brain is an extremely complex organ consisting of many regions. We look to focus on the motor cortex, which you can see in that first picture marked in red. This is the region of the brain which is really associated with muscular movement throughout the body. As you can see in the next image, the brain is covered by 5 main tissue layers. Studies have shown that the pia layer, which is the first covering the brain, can be practically removed from the brain without causing damage. The next is the arachnoid. This consists mainly of blood vessels and cerebral spinal fluid. Because of the makeup of these 2 layers and our desire to be as non-damaging as possible, we hope to place our chip beneath the dura mater within the arachnoid. The dura mater serves as an insulating protective layer for the brain and can be penetrated with low risk of injury. The pia and arachnoid layers offer negligible resistivity to the signals that we're trying to pick up with these chips and wirelessly put into prosthetic limbs. And due to this location, we would be able to avoid any surgeries into the actual brain itself.
Now that we know where to place our microelectrode array, we can speak about its design. What you see here is, first let me speak about what makes our design different from other people's designs. Okay? This is a 3-dimensional design. It's not as damaging to neural tissue as, say, like ECOG. And we call this Gandalf. Basically, Gandalf is going to be wired to an integrated circuit, which can then wirelessly take these neural signals and convey them to a robotic prosthetic. So let's begin with our fabrication process. We start with a silicon wafer that is coated with silicon dioxide. Well, from here, we add PDMS to it, a flexible, highly resistive, biocompatible siloxane polymer. So this is where we had to get creative, though, with our process. So we decided to make our electrodes first. We did this by placing SRS 100 on top of our PDMS. Upon developing, we were able to create a uniform array of 100 micron tall cylindrical electrodes. So now it's time we need to make our traces and our bump pads, so we put in SU810 in between our pillars. From here we have our chip that has our electrodes on it, and what we want to do here is we want to hit it with UV light. After we hit it with UV light, we're going to coat it with 15 nanometers of gold. Now what we found out here is that any part of this chip that is not exposed to UV light, those parts will be extruded from the chip. Finally, we separate our wafer from our PDMS, yielding our final product. Flexible, biocompatible, conductive neural probe. So before we continue, we have a little video showing our fabrication process.
Right here is where we coat the silicon wafer with PDMS before later house the electrodes. We then take it and place it onto a hot plate in order to make sure that the PDMS is cured. We now take SU8 and we place it on top of the PDMS bake it, and just to make sure they're hardened, and then expose it to UV light underneath the mask. After this, we bake it, and take it, place it within our heating developer, which is going to form our electrode pillars. And this we can view under your Hyrox 3D microscope. Do the same steps again to form the traces, but before we develop, we coat it with wool. Once coated, we have the Gandalf chip.
Now, what you have to understand about this chip is that our lab has been trying to create a chip like Gandalf for over 2 years. So imagine our surprise when just recently we were finally able to take these 2 years of research and turn them into a reality. And so that's what you see here. These are 4 experiments that contributed to this reality. In the top left, we needed to demonstrate that we could fabricate high aspect ratio pillars first before adding our traces. What you see over here, though, is that when we try to gold coat these pillars, we could only gold coat the top of these. And that was problematic for us, so we had to change our fabrication process. So down here on the bottom left is another demonstration that we could put our pillars first, and then build our trace pads and traces and bump pads, which is the reverse of the previous fabrication process. And then lastly, we needed to demonstrate that we could then